Oxaloacetate, that's a 5-carbon unit and gives you, is a precursor for all these amino acids. This is a little more complicated. Let's start with aspartate, asparagine, and threonine, which are shown here. Aspartate, threonine, what was the other one? Asparagine, that will be the end here. Okay, so they do come, and isoleucine eventually too. And you look at them and you kind of see the point. I mean, there's the 5-carbon skeleton here. You know, you have the keto acid, and here you have the amino acid, just an amino group different, and you can see the similarities as you go on and on and on. Okay, so aspartate to asparagine is just an extra amination here at the carboxyl group. So those are not, they don't seem too terribly difficult, and these are the carbons that are highlighted. So you can kind of see how oxaloacetate could be a precursor for all these things. A transamination is straightforward here, the alpha keto acid to alpha amino acid. Okay, you have an aspartate transaminase, like we have the glutamate transaminases. Here you have to have an extra amination to get to asparagine, and you know, these are a little more tricky, but they work. How do we go from homocysteine, let's say, to threonine, or homocysteine to homocysteine and methionine? And once you get to homocysteine, okay, this is again an amino acid that is not found. It is not found in proteins, but it's extremely important in metabolism. It's a natural amino acid. It's extremely important because it gives rise both to methionine and cysteine. Note here that you will see the structure in a minute, that the cysteine is actually produced, the 3-carbon of cysteine come from serine, because serine comes into this pathway, reacts with homocysteine, gives you a bigger compound, and then this part is actually lost, and what's left is the serine part plus the sulfhydrate here. So that's why the 3 carbons of serine give rise to cysteine. From threonine, you can go to isoleucine. Isoleucine is a bigger thing. You need to have 2 extra carbons. All right, so let's start from the beginning. Oxaloacetate to aspartate, you have a transamination. We don't need to discuss this again. And from aspartate, you can actually get to a lot of other amino acids. For example, aspartate is used here too in the urea cycle. Sorry, I don't know why. Okay, here. Where is aspartate? There it is. There is aspartate coming into the urea cycle to combine with citrulline and give you arginine and succinate. So aspartate has a lot of uses. From going from homocysteine to threonine, that's straightforward. You can dehydrate, and so you have a double bond, and you hydrate, and then you put the hydroxyl on the next carbon. So that can easily happen as well. So that's not a problem. From threonine to isoleucine, you need to increase the carbons, right? 3 carbons, but 2 carbons. Sorry, here, 2 carbons difference, right? So that's it. What's the source of those extra carbons? The source is actually pyruvate. So here we have, making isoleucine, you have inputs both from the TCA cycle in the form of the 4-carbon unit, and pyruvate, which is a glycolytic product, right? So both TCA cycle and glycolysis are needed to give you isoleucine. So they serve as building blocks, I should say, the intermediates in the TCA cycle and glycolysis to give you isoleucine. And there is something else that's a common theme here. Okay, I don't mention this slide, but we'll say it. So you put 3 carbons in to end up with 2 extra, so you lose a CO2. Why do we do that? So we make a carbon-carbon bond, and for that to happen, we have to lose 1 carbon. We've seen this principle again in fatty acid biosynthesis, when we have malonyl-CoA, being the building block actually, so that the chain in the fatty acid can extend by 2 carbons at a time. Well, malonyl has 3 carbons, but 1 carbon is lost to CO2, so you can drive the equilibrium through mass action, because it's a gas, it goes away, so you can drive formation of the carbon-carbon bond, which is normally unfavorable. All right, from homocysteine now to homocysteine, and we can get to these products. So we go from right to left. This is homocysteine. First, you acetylate it. In most species, is acetyl-CoA. Sometimes you use succinyl-CoA. It doesn't matter. You make this intermediate O-acetyl homocysteine, and then this is where sulfur. So there is no sulfur yet. Okay. The sulfur comes in the organisms that can do this reaction. As I said, we cannot do it. Okay? We take our methionine straight in the diet from organisms that can do this reaction. And H2S, so hydrogen sulfide, is where sulfur is how sulfur gets assimilated. It's an essential step. If you go back to the previous lecture, hydrogen sulfide, after you start from sulfates, you eventually reduce and you get down to hydrogen sulfide, and now it can be assimilated in this reaction to make you homocysteine. And that was the last reaction here in the sulfur assimilation that allows you to generate hydrogen sulfide with an enormous amount of reducing power. All right, so then you get homocysteine. Okay, so from homocysteine you get homocysteine, and this is a key intermediate because, as I said previously, you can go from here, you can go to methionine, or you can go from cysteine. These are the two sulfur-containing amino acids in proteins, cysteine and methionine. And from homocysteine to go to methionine, it's a single step. Okay, we were at this step before, and it uses something we have not seen before, a 1 carbon unit. So it gets an extra carbon, and that carbon comes from folates. Folates are this class of super important metabolites, essential for life. I mean, folate supplementation has been done on a public health basis since the 20s, especially for pregnant women. They're important for the development of the fetus. And once you supplement with folates, the incidence of failure for neural tube closure was diminished greatly. We fortify, that's the reason since the 90s that officially, many staple foods, such as flour, for example, rice, are fortified. When you hear fortified flour, it means fortified with these compounds, with these folates. What do they look like? The active business part looks like this. That's tetrahydrofolate. And this at this chain, it can be glutamated, but that's irrelevant here. So this is the business part. And they're really extremely useful 1 carbon transfer compounds. So this position 5 can receive the 1 carbon. Okay, so you can have the methyl tetrahydrofolate in this form. Instead of methyl, you can have other forms of carbon. It can be formulated, it can be methylene group. Okay, so they are the universal, let's say they're excellent receivers of 1 carbon units, and they can pass them on to other compounds, such as homocysteine, which looks like this. And the difference between homocysteine and methionine is that the sulfhydryl group is methylated in methionine. So it's that extra methyl group that makes all the difference. And this is where the folates become very important. All right. So the source of carbon for the methylation of homocysteine to give methionine is folates. And the enzymes that do this also have a vitamin B12 as a cofactor. Vitamin B12 is a very complex. It looks like this. Okay, so it's super complex. So the chemistry is fascinating. But that brings us to the methyl cycle. All right, so we got from homocysteine, we got to methionine, as we discussed with tetrahydrofolate. And that gets regenerated in a variety of other ways, which we will see. By the way, these reactions are very important for also folate-based reactions are essential for making nucleotides. So we'll see those in a couple slides, in a couple lectures. And in fact, interfering with this folate cycle with inhibitors, the anti-folates, is the staple of anti-cancer therapy. But that's for a couple lectures later. For now, we start with homocysteine. We saw how that was made, and we got methionine, our first sulfur amino acid, right? Now, methionine is also a major donor of methyl groups. It is actually the form of, it provides it is a source, I should say, of methylations for a variety of things in the cell. And if you ask how important are methylations, by far one of the major sinks of methylations in the cell is to methylate our genome. Okay, histones get methylated. In humans, DNA, the bases are also methylated. And for all these reactions, the methyl groups come ultimately from methionine. How does that happen? So methionine, right here, generates, it gets phosphorylated. So this sulfur group, it gets attacked, and it gets, you have S-adenosyl methionine. So in the whole, this part of ATP without the phosphates comes in, okay, and sticks itself to the sulfur. And of course, you lose pyrophosphate and phosphate. So you consume all of the ATP here. But this part, the S-adenosyl methionine, is now the universal, can now transfer this methyl group onto a variety of substrates. It could be proteins, it could be other things, it could be DNA, and X, that's the X. And what you generate in the end is S-adenosyl homocysteine, you know? So it's really homocysteine plus the adenosine part. And you can hydrolyze this and regenerate homocysteine and keep doing this over and over and over again, right? Provided that you bring in 1 carbon units to the system through folates, okay? So all the 1 carbon input comes through folates here, and the output is all the methylations, or one output is all the methylations. And we'll see how the 1 carbon input is generated onto folates. The hint, it comes from serine, okay? But we'll see that. All right, so we saw how we go from homocysteine to methionine. How now do we go from homocysteine to cysteine? This is this part down here. We saw this part of the slide. Now it's pretty straightforward. Serine will react with homocysteine, will make cystathionine. Okay, so this dipeptide in a sense. And then you remove one amino group and you got this, and you hydrolyze, sorry, and you remove the amino group and you got cysteine and alpha ketobutyrate, which you can use and convert to propionyl CoA. And that's how we got cysteine. Now, this part of the reaction is actually extremely important because cysteine finds itself not only in proteins, big proteins, as a sulfur-containing amino acid, but in small peptides such as glutathione, which is a tripeptide that it's extremely important for the cell to mount it has antioxidant properties. So that it makes, whether the cysteine, the sulfhydryls are reduced or oxidized, they can be used, okay, to detox, to re-reduce, let's say, things that have been oxidized inappropriately. So there is a lot of flux that goes into generation of cysteine for glutathione. Now, importantly here, the carbons that all the carbons that are found on cysteine came from serine originally, right? Okay. Oh, and the enzyme that does this reaction it's a special enzyme called the cystathionine beta synthase. It's actually quite important. It's the #1 since we're not doing presentations anymore. It predisposes people to atherosclerosis if there are mutations in this enzyme. And these individuals have super high levels of the substrate, which is homocysteine, and you have this situation where you have homocysteinuria, and that's toxic.